can't see the slides.
Excellent, excellent. So sorry, I was supposed to give this first presentation yesterday, but we moved this talk to have it together with the gene therapy presentation. So thank you. First of all, thank you for the invitation to be with you today. It's a pleasure to be. Unfortunately, we are not able to be face to face, but you know, the technology allows us to communicate. So thank you. I will start with the first talk about motor neuron disease, and I will focus mainly on amyotrophic lateral sclerosis and give you an overview about the disease, some insights in genetics. So that basically, these slides outline what I will be discussing in my first presentation, what is ALS, the classification and pathogenesis of ALS, and insights from genetics. So basically ALS is one of the devastating neurological disorders, and probably if you talk to neurologists, probably they consider it one of the worst diseases that affect the nervous system. It's the commonest adult onset. Most of the symptoms appear in adult individuals, and the incidence is around 1 to 2 per 100,000, and it seems that it's rising. It's a progressive degeneration of upper and lower motor neuron, and next slide explain what is upper and lower motor neuron. And you can see here the upper motor neurons the cell bodies are located in the cortex, and the axons are basically going all the way down to the spinal cord to connect to other neurons in the spinal cord. The lower motor neurons, there are different categories here. So you can see here in the brainstem, innervating muscles in the face, for example, and other part of the face, but also motor neurons emerging from different part of the lumbar spinal cord and innervating limbs and other muscles, intercostal muscles, for example. So this is just another illustration of upper and lower motor neurons. So you can see this layer of the layer 5 of the cortex, where cell bodies of the upper motor neurons are located, and lower motor neurons, as I said, they are located in the ventral horn of the spinal cord, for example, here. And you can see this beautiful motor neurons of the spinal cord. When these motor neurons degenerate, they lead to a number of symptoms, and you can see here the major atrophy of muscles in the limbs, legs, and hands as well. And these are the dysfunction that is related to lower motor neurons. In terms of classification of ALS, so there are variants of ALS. So you can see here, depend on if lower or upper or mixed. So you can see, for example, the primary muscular atrophy, it's only lower motor neurons that are affected, while primary lateral sclerosis, upper motor neurons are affected. And when we have both type of motor neurons affected, this results in ALS. In some situations, there is overlapping phenotypes, and as you can see here. So ALS is, as I said at the beginning, is a devastating disorder, and the reason for that, in most of the cases, it's very fast. The progression of the disease it takes 2 to 4, 5 years after the first diagnosis for patients to die. And the majority of these patients are classified in subgroup with rapid progression, as I said, with the onset older age and mostly bulbar onset. I will explain this later on in terms of the different type of bulbar limb and respiratory onset. In some cases, very rare, patients, they can survive and live over 10 years. And we have examples of this in many occasions. As I said, the incidence is 1 to 2 per 100%. Most of the cases are sporadic, but familial cases exist in 5 to 10% of cases. These are the different type of onset, focal onset, and I will start with the limb onset. Basically, this is the commonest pattern. So this exists in most of ALS patients, and the symptoms or the atrophy and weakness in muscles appear first in limbs, arms, legs, and they start with a single limb first, and usually it's asymmetrical. So, and then spread to the other side. The bulbar onset usually is characterized by difficulty in speaking clearly and swallowing. So the symptoms start around this area of the body, and it's reported in around 20 to 25% of ALS cases. For some reason, we don't know why it's preponderance in female versus male. The last type of onset is respiratory onset, and these cases are considered rare among the ALS cases, and it's characterized by the onset. The first onset appear in weakness in the intercostal muscles that are helping basically with the breathing, and diaphragm as well is involved in breathing, and it's triggered by the degeneration of motor neurons located in this part of the spinal cord. These are some characteristic symptoms: the hypoventilation, and patients frequently complaining by frequent awakening and early morning headaches as well. This is just to illustrate the difference between a normal spinal cord. These are sections, and you can clearly see ALS spinal cord. There is, major morphological changes here. Morphological changes and the pathology also is characterized by protein inclusions. And here are some examples. So you can see ubiquitin positive taking these shapes dispersed in the cells in motor neurons, for example. But also we have this ubiquitinated inclusions that are compact in the cell body. You can see them here. In terms of genetics of ALS, as I said before, the majority of cases are sporadic, but 5 to 10% are familial. Many mutations, genes are involved in ALS, and overall over more than 10 known causative genes. Most of them are dominant, caused by autosomal dominant inheritance. Some very rare autosomal recessive as well were reported. So the first gene that was discovered is the superoxide dismutase 1, SOD1, and this is the first that was linked to ALS patients, dominant mutations in this gene, and it's causing around, on average, 2% of the total cases. Recently, a more interesting gene was discovered, reported to cause ALS, and it's the most common gene, causative gene in ALS, around 40% in familial cases, around 7% is sporadic. And this is the C9orf72. Obviously, there are other genes like TARS-BP, which is TDP43, FAS. They are RNA binding proteins, and they are linked to ALS. There are other genes that were linked, but the number or they are quite rare in cases. As I said, the first gene was SOD1 superoxide dismutase. This is a gene encoding a cytosolic antioxidant enzyme, SOD1. And this enzyme is helping to clear superoxide radicals from cells. It's expressed, ubiquitously expressed, and it's a homodimer. It does contain copper and zinc atoms in active sites. So in terms of mutations linked to SOD1, the majority are autosomal dominant with adult onset disease. Some cases of autosomal recessive inheritance, but they are quite rare. In particular, this D90A mutation in Scandinavian patients. The characteristic of SOD1 mutations, it leads to toxicity or cell death by a toxic gain of function, and there is no correlation between enzyme activity and severity of disease. And major discoveries actually in ALS were made in this transgenic animals, mice with different mutations, and the most common and widely used one is G93A mutation in mice, and these mice develop symptoms similar to ALS patients. The mechanism of action, it's widely debated. Obviously, there are some reports of copper and zinc toxicity, abnormal free radical handling, protein aggregation is widely reported, linked to SOD1 mutations as well. The second gene is, as I said, is TARP-BP, TDP43, and this has been identified as a component of ubiquitinated protein inclusions in both sporadic and familial ALS. And TDP43 is expressed, a protein expressed in everywhere, in all different cell types, but primarily localized in the nucleus. It's involved in RNA processing. It has been reported that it's interacting also with SMN, survival motor neuron, the gene involved in spinal muscular atrophy. This is the childhood form of motor neuron disease. I will talk about that later on in my second talk. And it has been also reported that it's TDP43 stabilizes neurofilament, in particular neurofilament L mRNA. The mutations in TDP43 or TARP-BP gene were reported originally in two large familial ALS pedigrees and two sporadic ALS cases, and that was in 2008. There are more than 35, and I think there are more than that actually now. I need to update this slide. The third gene, FAS-TLS, so this is fused in malignant sarcoma, FAS gene identified in 2009. And again, the majority of cases reported are mutations are autosomal dominant with adult onset, although some rare families show autosomal recessive inheritance as well, as you can see here. So, like TDP and SOD1, FAS is ubiquitously expressed primarily in the nucleus. It's an RNA binding protein, and it helps shuttles RNA between the nucleus and the cytoplasm. The most recent gene linked to ILS is C9orf72, and this is quite an interesting gene because it's linked to this expanded G4C2 repeat expansion. And this repeat expansion exists in the intron. And it's interesting because it has been reported to in a large number of familial cases, as you can see here, but also as a significant number of sporadic ILS cases. It's also linked to FTD, both familial and sporadic FTD, as you can see here. So, as I said, the expansion, GC-rich expansion is located in the intron 1. And what this does is that it leads to the production of this RNA foci that report to, there are some hypotheses that it leads to toxicity in the cells. But the most relevant one and widely reported that leading to toxicity is the production of these dipeptides, DPRs. And both sense and antisense dipeptides can be produced through RAN translation. So this is again the repeat of some of what I said in previous slides, just summarizing different possibilities of the mechanism how C9orf72 repeat expansions lead to cell death, like a loss of protein function, although that is debated. RNA toxicity, I mentioned these foci that are forming mainly in the nucleus, you can see them here. And the dipeptides production, both sense and antisense. And finally, some hypotheses, and we in Sheffield did some work on this with Sharif Qasimi. So we showed that C9orf expression is linked to the DNA damage. So this is just a summary of what I said in my slides, and I will not go through this. As I also mentioned that mainly SOD1 mutations were introduced in different model systems, including fly, Drosophila, and zebrafish mouse model. The widely used animal model is mouse G93A, although other mouse models are also used to study mechanism, but also to test therapeutics. ILS is a complicated disease, and different mechanism and hypotheses are put forward to explain the mechanism of toxicity in motor neurons, but also in other cells, because glia cells, astrocytes, are reported also to have some abnormality in ILS. So this is just a slide summarizing that with different hypotheses: mitochondrial dysfunction, apoptosis, excitotoxicity as well, protein misfolding, and impaired axonal transport. But probably this mechanism are relevant, all these mechanisms are relevant and lead together to motor neuron degeneration in ALS. So I will stop here.